You're listening to Agriculture Today from the campus of Kansas State University. Every once in a while, as we visit about scientific advances in agriculture, we'll come across something that may truly be regarded as a breakthrough. And that's what we'll discuss right here. For scientists here at K-State have possibly found an answer to our issues with herbicide resistance in weed populations. This could be a great leap forward in that field. Joining us now is a weed scientist from the K-State Department of Agronomy, Mithila Jugulam, and along with her is a plant geneticist at K-State, Bikram Gill, and they were co-authors on this research. Mithila, we'll bring you in first of all, and this is really in your area as a weed scientist, resistance to herbicides in weed populations. So this has been worked on a long time, hasn't it?
Yes. The herbicide resistance in weeds has been a huge problem, not only in Kansas, throughout the U.S. and also many parts of the world. But in this research, essentially what we looked at was glyphosate, which is globally widely used herbicide, resistance in our pigweeds, which are palmer amaranth and water hemp. So what is new that we found in this research was how these weeds have evolved resistance to glyphosate in such a short time. Mm-hmm. Because if you look at the evolution of glyphosate resistance in palmer amaranth or water hemp, it occurred, based on our research, it appears to occur very rapidly. So that is one big news from this research. The other important aspect of this research was that these resistant plants carry the herbicide glyphosate target gene in humongous numbers. In the sense, they have massive copies of this target gene. Therefore, when you apply even like 10 times recommended dose of glyphosate, the plants will not be killed. That is a very fascinating finding as well. Another important aspect of this these copies that I mentioned appear to present in the form of a circular chromosome structure. So, which is what is so novel about this finding.
That may well be what you call, Bic, the ring chromosome, the secret at hand. This has been hard to find or discover over time. Is that correct?
Yeah, so normally our genetic material in all organisms, including us, is found in these linear chromosomes, long linear DNA molecules. And the herbicide target gene is also found in a linear chromosome. But it looks like when we looked at these glyphosate-resistant weeds, the gene actually escaped from the chromosome and it formed a circular chromosome. And then that circular chromosome can amplify very rapidly, so it found this as a mechanism to amplify in large numbers so that the enzyme that this gene produces, it can detoxify glyphosate and develop resistance.
So in identifying this circular chromosome then, what could that potentially allow researchers in as far as developing an answer to this resistance problem?
So the circular chromosome, the way it transmits to the germ cells is through some unknown tethering mechanism. We saw that these little circular chromosomes, they piggyback onto the normal chromosome during cell division or during meiosis, and they transmit to the gametes and to the next generation. So knowing this, if we can understand this mechanism, how they do it, there might be some solution to this problem, some strategies.
What, as you, Mithila, look at that, would be some strategies that you might be pursuing in further research? Somehow dealing with this particular chromosome, this ring chromosome, and removing it from the picture, so to say? What?
Actually, in the literature, it's been known that these ring structures could be unstable. So what we want to explore is, for example, if we do not apply glyphosate repeatedly or reduce the selection by glyphosate, can we make these ring-structured chromosomes unstable and eventually make these plants susceptible to glyphosate so that it could be still an option for us to manage these weeds? That's one thing we want to explore in the near future. And also, if you look at advanced technology, once we know the gene sequences by which these ring structures are attached to the linear chromosomes, can we kind of silence that gene or the nucleotide sequence and make them sensitive? That's kind of really a long-term research. But to start with, like I mentioned, we want to see if taking the herbicide pressure off, can we make these copies unstable and see if that could be a potential means of controlling these weeds.
Yeah, that's a good point. These circular chromosomes, they are unstable. So if there's no selection pressure, the theory is that they will disappear. So if that happens, that means there might be something in terms of management. One very important point is we think that these ring chromosome resistance is transitory. So we have somehow caught it in between becoming permanently resistant. So eventually we saw that these ring chromosomes are incorporated into the linear chromosome, the normal chromosome. And if that happens and then they become selected, then it'll be permanent, then it'll be resistant forever. So it's possible that this is just an intermediate stage because it's a very rare event and heavy selection pressure probably selected this plant with a ring chromosome. But we think it's the intermediate stage. It's on its way to becoming highly resistant. So if you reduce the selection pressure, we think ring chromosome will disappear. And
yeah, it could be like still, we can make these weeds susceptible.
So that's the experiment we have to do.
Yeah, that's our next research area of research that we are interested to look at. Yeah.
Now, your work, as you noted, concentrated on Palmer amaranth and water hemp, two huge weed issues here in Kansas. Can this same theorem, if you will, apply to other weed types?
Potentially. We have some evidence also. If you look at Palmer amaranth or Amaranthaceae weeds, they are broadleaf weeds. As growers know very well, they are dealing with both broadleaf and grass weed issues. Although not found yet in Kansas, we have worked with a grass weed, which is ryegrass, resistant to glyphosate, in collaboration with the University of Arkansas. So in that particular grass weed also, we have convincing data to say that potentially ring chromosomes are present that carry these glyphosate target genes as well. So it seems like it's not specific to broadleaf weeds or it's across the board.
Yeah. Actually, the circular chromosome or ring chromosome have been recently, last 2 years, they have found them in yeast. They have found them that they are also involved in cancer. 80% of the cancer cells cause cancer because these genes, the ring chromosomes, are amplified and they lead to uncontrolled cell growth. So they seem to be involved in all kinds of sort of resistance to drugs, or where a plant is stressed, the cell fights that stress through these ring chromosomes. So this is, you'll be hearing a lot an overview about these ring chromosomes, and this is a new advance in plant genetics, and we're just trying to figure out, how many different things and how many different organisms they are involved. I think this is just a tip of the iceberg.
And that is why you have stated in remarking on this work, Bick, that this may well be the most significant plant genetic advance in your long and accomplished career at Kansas State University.
Yeah, this is very exciting. I have spent all my life working on wheat, bread wheat, where we defined all these technologies. And when we started working with Mithila on a weed, we said, my God, all our genetics we have learned is going out of the window when we start looking at these elements. So it was very, very exciting. It just tells you that, if you can work together and bring your different tools together, you can just do some wonderful science.
Yeah. Mithila, you're obviously excited about where this is
I'm
going.
excited about this, and it's so fascinating to work with weeds. Honestly, the mechanisms are all in weeds' genetic diversity. That's where it's coming from. So it's all naturally evolved. Like we are seeing this happening, evolution in action, basically. This is what it is.
This is a huge discovery, but you've work to do. You're not done yet, right?
No, just the beginning. We have got all
beginning.
these ideas now to explore and work with.
Yeah, how the cell ring chromosome replicate, how they do all the kind of stuff. A lot of basic knowledge we need to learn about.
And we wish you both well in continuing this work, for what you've uncovered here may well set the tone for a permanent answer to herbicide resistance in weed populations out there. So congratulations once again, and thank you for sharing in brief the story of this work right here.
Well,
Thank you.
Eric.
Joining us here on Agriculture Today, Mithila Jugulam. She is a weed scientist at K-State, and Bikram Gill, a plant geneticist here at the university. Once again, they've identified what they're calling a ring chromosome, which may be the catalyst for weed herbicide resistance. And the potential of manipulating that catalyst could lead to answers to that question of resistance in weed populations. You're listening to Agriculture Today. We'll be back in a few moments on the K-State Radio Network.
Agriculture and food systems are the main drivers of the Kansas economy, but must be improved in order to feed the world's growing population. How are we going to do so? Reduce food loss, find ways to preserve grasslands, and help families stretch their dollars. Global food systems is one of the 5 grand challenges K-State Research and Extension is addressing. To learn more, visit www.ksre.ksu.edu.
We're back now on this Agriculture Today, and we are close now to the beginning of the traditional pasture burning season in Kansas, the prescribed burns that are a routine of management for our grasslands. We say that because there are a pair of special workshops coming up for producers who do conduct prescribed burning on smoke management. These are set for later this month in one case and very early April for the other. K-State is hosting these, of course, and along with us, a biological and agricultural engineer here at K-State, Zifei Liu, who not only is going to be organizing these workshops and conducting them, but has been deeply involved in researching the behavior and the makeup of smoke from our prescribed burns. And Zifei, you might talk with us a bit about your work before we get into the workshops and their purpose.
Okay. First of all, I am an air quality specialist at K-State, and I have been conducting research on smoke impact of rangeland burning since 2014 when I joined K-State. And I have been providing smoke management workshops since 2015. And for the past 3 years, I provide the workshop annually before, during the burning season. And the number of audience is growing for the past 3 years. So this year, I decided to do 2 sessions, and one in Manhattan and another in Emporia, so that we can allow more participants.
And those are taking place, by the way, on Wednesday, March the 21st in the case of the Emporia workshop, Tuesday, April the 3rd for the Manhattan workshop. And you will be sharing information from your research on the behavior of smoke and the contents of smoke. What is the intent here?
Intent, yeah. We know that prescribed burning is a long-lasting practice to maintain the ecosystem here. There is a wide consensus that the burning is necessary to protect the ecosystem, but there is different opinion on when to burn and how frequent we should burn, and also there is a lot of intention or discussion about the smoke impact of the burning, because the burning generates smoke, and the smoke can impact large area downwind communities, and also may contribute to the exceedance of the current air quality standard. Mm-hmm. And as we know, the air quality standard is evolving and has a tendency to be strict, and especially the ozone. We know ozone is a very dangerous air pollutant, and unfortunately in Kansas we have a high background of ozone, and it's not easy to keep the ozone under the standard. So there is a lot of evidence that smoke contributes to high level of ozone. So what we intend to do is try to generate a smoke management plan, help to reduce the impact on ozone.
And that's what you've been researching, the elements that you just described that lead to the ozone buildup, and whether or not prescribed burning can be managed in such a way to minimize the potential of that impact?
Yes.
And what ways have you come up with so far in that respect? Timing of the burn, paying attention to the conditions of the designated burning day? What?
Yeah, because open burning is hard to put a control device, right, like what we do to other pollution source. Management is the key. So timing is a very important strategy. When we making burning decisions, usually it's driven by different management objectives. Also, fire safety is the major concern to decide whether to burn or not. And KDHE and K-State have already developed a smoke management tool to help land managers to timing their burn, also consider the smoke dispersion, and help them to plan the burn, timing the burn to avoid effect on sensitive areas like big cities or high populated area. But currently, these smoke management tools are not capable to forecast the impact of ozone. So in these two workshops, I want to focus on the knowledge on ozone. Mm-hmm. We want to establish the relationship between fire activities and the ozone level, use the historic data.
Are you hoping, before we talk more about the workshop, but as you further this research, are you hoping to devise some way of accurately forecasting the ozone then? Is that part of your objective?
Yeah, the ultimate goal is to generate a practical tool which can help us to better distribute the burning activities and help us to better timing of the burning. Before we do that, we want to generate some knowledge, make the connection between the fire activity and ozone. Ultimately, there is a lot of way to modeling ozone. For example, a photochemistry model. But photochemistry model is very expensive and need a very high requirement of input data, like how much is emitted and the photochemistry procedure, how ozone is formed. So currently, is still a long way to make it a practical tool. Mm-hmm. So what I want to do is generate a model based on the historical data, and we have a lot of historical data available now. Some of them are from satellite data available since 2000. And they accumulated a lot of data on fire activities. And also we accumulated a lot of ozone data. Ozone monitoring network provide a lot of data. So by analysis of this data, we may be able to generate a very clear relationship on how much the fire activity actually contributed to ozone and water conditions.
So that will be valuable information as it comes together. But for the present, there is the smoke management tool out there at ksfire.org, by the way, which you're encouraging producers to fully utilize as they're approaching prescribed burning time. But moreover, an understanding of what we know to date about smoke behavior, smoke content. That's the mission of these workshops, to share that information with producers, correct?
Yes. One objective of the workshop is continue to promote the current smoke management tool, which is available at ksfire.org. And that tool is very useful to reduce the smoke impact and help you to time the burn activities. Mm-hmm. Another objective of this workshop is to provide the knowledge currently obtained from the historical data about the impact of smoke on ozone levels.
Very good. So this is free and open to the public, you say, this workshop. And it's the same program at each location, correct?
Yeah, similar program at each location. And we wanted to be open to public because we think the communication is very important, because the smoke not only have environmental impact, also have a social impact, social intent. People need to know how smoke can affect my health. Also, I'm an air quality specialist. I'm a land management specialist. I also need a lot of input from different stakeholders so that we can share our vision how to keep this burning practice, but in a manner that minimize the impact, both environmental impact and the social impact.
So you're encouraging a dialogue. It's not just a one-way presentation.
Yes.
And how does one participate? Do you need pre-registrations?
I hope if I'm interested, please email me with your name. That's enough, so I can have a better head count. Yeah.
And his email, we'll repeat this, is Zifei Liu at ksu.edu. That's Zifeiliu at ksu.edu. But it is important to continue to exchange this information, not only to and with landowners who prescribe burn, but to even city dwellers, anybody interested in the topic of smoke emitting from prescribed burns should gain something from these meetings.
Yes, I try to update information every year, add something, and with my most recent research, and hopefully it will be helpful. Yeah.
And we wish you well with these. They're coming up very soon. And thank you for briefing us on not only your work, but what you hope to share from that work at these workshops coming up soon, Zifei. Good luck with these.
Thank you.
He's an air quality specialist out of the Biological and Agricultural Engineering Department at K-State, Zifei Liu. And again, the core of his research work has been on the nature, behavior, and content of smoke, which emits from our prescribed pasture burns here in Kansas. And these 2 prescribed burning smoke management workshops are set for Emporia, Wednesday, March the 21st, starting at 9 o'clock in the morning. The Emporia workshop taking place at the Boyer Community Building on the Lyon County Fairgrounds. And then in Manhattan, Tuesday, April the 3rd, at the Kansas Farm Bureau Building at the northwest edge of Manhattan. And that'll start at 1:30 in the afternoon. The workshop's running roughly 2 hours each. Same program at each location. Do email Zifei to reserve your spot so they can make the proper accommodations and prepare the materials for these workshops. Zifeiliu at ksu.edu. Zifei Liu at ksu.edu. And we've referenced it a time or two in our visit here, but once more, pasture managers do make full use of that prescribed burning smoke management website. You can obtain information and access those tools to assist you in making burn decisions based on the smoke management plan. The featured element of that website is the forecast map and the smoke modeling tool, the map showing the potential smoke impact of burning in your area on air quality downwind. It is updated daily, and the modeling tool allows you to see where that smoke will go from a fire in your location. Both the forecast map and the modeling tools are available on mobile devices for your convenience. All of that is found at ksfire.org. Once more, ksfire.org. We'll be back with more on Agriculture Today after this break. You're listening to the K-State Radio Network.
Did you know every Kansas farmer feeds 128-plus people?
Kansas farmers are hard workers, dependable, authentic, and sensitive.
Not only do farmers put food on your table, but they put clothes on your back and fuel in your car.
For more information about Kansas farmers, visit K-State Research and Extension online or stop by your local Extension office. This message has been brought to you by the K-State Animal Sciences Leadership Academy participants.
You're tuned in to Agriculture Today. For you now, our weekly wildlife segment. Charlie Lee is aboard once again, wildlife specialist, K-State Research and Extension. Charlie, a closer look this week at a pest that we find in row crop fields, especially no-till corn. That would be the vole. And as innocent as it might seem, this critter can cause some serious crop damage.
Yes, prairie voles are a species that can get quite numerous. And now is the time to think about how to manage your vole populations if you are in an area that has problems with voles. The mature prairie vole is a reddish brown to gray in color. It's a little larger than a field mouse, but smaller than a rat. They have a tail that's about 1.5 inches long, probably about 1.5 times the size of a mouse would be the size of a prairie vole. But I think most people would recognize the voles by the trails that they leave behind in areas that have a lot of decaying litter on the surface. And when you think about no-till fields with a lot of crop residue on the surface, that makes ideal habitat for voles. And under ideal conditions, voles are going to produce a litter about every 21 to 26 days. They don't reproduce every month of the year, but probably 8 to 10 months of the year are going to have a litter of young. So populations can grow very rapidly, and consequently, they are going to feed on whatever is available.
And that could be, in this instance, newly planted crop seed.
Yes, they have very broad food habits. They will consume seeds, newly planted or waste grains in the field. They're grass seeds, weed seeds, herbaceous material like young growing green crops, and other habitats like around a homestead. They may feed on bark of shrubs or even mature trees, whether it's deciduous or coniferous trees. So they may burrow under the soil surface, but most of the time they form their colonies on top of the ground underneath decaying leaf litter. And at times we've measured populations of voles of up to 500 per acre in ungrazed, unmanaged rangeland situations. And it's due to having these ideal habitat conditions with that decaying leaf litter on the surface that protects them from overhead predators. Typically, we see a pattern of the population growing for 2 to 3 years, and then we get a crash and simply don't have that particular problem anymore. The lifespan of a vole is not very long. Typically, it's going to be less than a year and a half, and about 80% of those young may die if the habitat conditions are not good. But what I see as these problems become more and more prevalent across the state is that conservation till and no-till provides ideal vole habitat.
And that, as proven by research, can amount to substantial crop loss in the case of corn anyway.
Yes, it's not that they're going to damage the mature crop. The damage comes because they're digging up the newly planted seeds, which may wreck the plant population. Voles have a pretty sensitive nose. They're able to grow right down the row and make a small hole and dig up each individual seed. After about 2 to 3 weeks time frame after planting, the seed itself is used up as in the production of the plant. The voles then simply start feeding on waste grain or even on large amounts of cutworms and wireworms, grasshoppers, weed seeds, and other things that may be left in the field. So it's a very short time frame when they can be detrimental to your crop, but it is a time that they can have significant damage, and it's been measured of over 100 bushels per acre yield loss in those areas that are inhabited by vole colonies.
Well, that should get one's attention right there. So control options here, and you point out, first of all, that you need to, before you act any further, confirm the presence of voles by scouting.
Yeah, scouting is certainly important. It needs to be done about 30 days before planting. Usually occurs in the first part of March. Look for active vole colonies. They're delineated by these runs, which are narrow trails about the width of a bicycle tire track in fields that have good aeration. If there's vegetation present, you'll often see nice green spots. That's often areas of vegetation, grass that may be coming up, annual grasses, fertilized by the urine and feces of that high number of voles in that particular area. That delineates the vole colony. You'll see openings to their burrows about the diameter of a golf ball. Some of those burrows will go underground. Some will just be under the vegetation that's found on the soil surface. So when you find more than 5 of those vole colonies per acre, we think that that's kind of the threshold. You should start seriously considering vole control when populations reach that level.
And that control would be in the form of a toxicant application then?
Well, there's several other options, but toxicant is what most people will want to go for. But I'd encourage them to think about some of the other options. And one of those is just simply cultural control. Can you reduce that food and cover by mowing? Can you do it by cutting a little closer to the ground and shredding that residue after it goes through the combine? Whatever you can do to reduce the amount of vegetation that's found on the soil surface is going to do a good job at preventing those problems. But once you have the problem, then toxicant is probably going to be the technique to use. Unfortunately, we don't have very many that are labeled for voles, and we have even fewer toxicants that are labeled for voles in crop fields. The only product that we would have would be zinc phosphide. It does have a label for no-till corn, but it's designed for in-furrow application at planting or pre-planting. So time is of the essence for those people that are having problems with voles in no-till corn. Do a good job of going out and scouting. If you are finding more than 5 vole colonies per acre, consider an in-furrow application pre-plant in order to get those populations down to a manageable level.
Charlie, we appreciate the word on voles and the damage they can inflict on particularly no-till corn production. Charlie Lee, wildlife specialist, K-State Research and Extension. He's along with us every Tuesday right here. Our time is away for today. We appreciate you being along with us and hope you'll rejoin us right here tomorrow, same time. Until then, Eric Atkinson here. This has been Agriculture Today over the K-State Radio Network.